Now we're going to learn some selected topics on the cellular and molecular basis of immunity, only some glimpses into some major topics. So first we're going to learn the molecular structure of antibodies. Antibodies are proteins produced by B cells and their descendants. Descendants in the sense plasma cells. So B cell incorporate antibody molecules into their plasma membrane where they serve as antigen receptors, whereas plasma cells secrete these proteins into the blood or other bodily fluids where they serve as molecular arsenal in the body's war against invading pathogens. So interaction between blood-borne antibodies and antigens on the surface of a virus or bacterial cell can neutralize the pathogen's ability to infect a host cell and facilitate the pathogen's injection and digestion by wandering phagocyte. So we just learned antibodies are proteins produced by B cells and their descendants. Descendants in the sense plasma cells. So B cells incorporate antibody molecules into their plasma membrane where they serve as antigen receptors, whereas plasma cells secrete these proteins into the blood or other bodily fluids where they serve as a molecular arsenal in the body's war against invading pathogens. Interaction between blood-borne antibodies and antigens on the surface of a virus or bacterial cell can neutralize the pathogen's ability to infect a host cell and facilitate the pathogen's ingestion and destruction by wandering phagocytes. So the immune system produces millions of different antibody molecules that taken together can bind any type of foreign substance to which the body may be exposed. Through the immune system exhibits great diversity through the antibodies it produces. A single antibody molecule can interact with only one or a few closely related antigenic structures. So antibodies are globular proteins called immunoglobulins. Immunoglobulins are built of two types of polypeptide chains, larger heavy chains, molecular mass of 50,000 to 70,000 daltons, and smaller light chains, molecular mass of 23,000 daltons. The two types of chains are linked to one another in pairs of disulfide bonds. 5 different classes of immunoglobulin IgA, IgD, IgE, IgG and IgM have been identified. The different immunoglobulins appear at different times after exposure to a foreign substance and have different biological functions. IgM molecules are the first antibodies secreted by B cells. IgM molecules are the first antibodies secreted by B cells following stimulation by an antigen appearing in the blood after a lag of a few days. So IgM molecules have a relatively short half-life, about 5 days, and their appearance is followed by secretion of longer life IgG or IgE molecule. So IgG molecules are the predominant antibodies found in the blood and lymph during a secondary response to most antigens. So IgE molecules are produced at high levels in response to many parasitic infections. Then IgE molecules are also bound with high affinity to the surface of mast cells, triggering histamine release which cause inflammation and symptoms of allergy. So IgA is the predominant antibody in secretions of the respiratory, digestive and urogenital tracts and acts to protect these mucosal linings from pathogens. The function of IgD is unclear and that portion is always open for research. Then there are two types of light chains, kappa chains and lambda chains, both of which are present in the immunoglobulin of all 5 classes. In contrast, each immunoglobulin class has a unique heavy chain that defines that class. So we will focus primarily on the structure of IgGs. An IgG molecule is composed of 2 identical light chains and 2 identical heavy chains arranged to form a Y-shaped molecule. To determine the basis of antibody specificity, it was first necessary to determine the amino acid sequence of a number of specific antibodies. So normally the first step in amino acid sequencing is to purify the particular protein to be studied. Under normal conditions, however, it is impossible to obtain a purified preparation of a specific antibody from the blood because each person produces a large number of different antibody molecules that are too similar in structure to be separated from one another. The problem was solved when it was discovered that the blood of patients suffering from a type of lymphoid cancer called multiple myeloma combined large quantities of a single antibody molecule. So, cancer is a monoclonal disease, that is, the cells of a tumor arise from the proliferation of a single reward cell. Because of a single lymphocyte normally produces only a single species of antibody, a patient with multiple myeloma produces large amount of the specific antibody that is synthesized by the particular cell that becomes malignant. One patient produces one highly abundant antibody species and other patients produce different antibodies. As a result, investigators were able to purify substantial quantities of several antibodies from a number of patients and compare their amino acid sequences. An important pattern was soon revealed. It was found that half of each kappa light chain has a constant amino acid sequence among all kappa chains, whereas the other half varies from a patient to patient. A similar comparison of amino acid sequence of several lambda chains from different patients revealed that they too consist of a section of constant sequence and a section whose sequence varies from one immunoglobulin to the next. The heavy chains of the purified IgGs also contain a variable V and a constant C portion. The schematic structure is given. So disulfide bond is there, COOH portion along with Cl domain and Vl domain will be there. Antibody domains. Okay. It was further found that whereas approximately half of each light chain consists of a variable region VL, only 1/4 of each heavy chain is variable VH among different patients. The remaining 3/4 of the heavy chain CH is constant for all IgGs. The constant portion of the heavy chain can be divided into three sections of approximately equal lengths that are clearly homologous to one another. These homologous Ig units are designated CH1, CH2 and CH3. So it appears that the three sections of the C parts of the IgG heavy chain as well as those of heavy chains of the other Ig classes and the C portions of both kappa and lambda light chains arose during evolution by the duplication of an ancestral gene that coded for an Ig unit of approximately 110 amino acids. The variable regions VH or VL are also brought to have arisen by evolution from the same ancestral Ig unit. So structural analysis indicates that each of the homologous Ig units of a light or heavy chain folds independently to form a compact domain that is held together by a disulfide bond. So in an intact IgG molecule, each light chain domain associates with a heavy chain domain. So genetic analysis indicates that each domain is encoded by its own exon. So the specificity of an antibody is determined by the amino acids of the antigen combining sites at the ends of each arm of the Y-shaped antibody molecule. The two combining sites of a single IgG molecule are identical and each is formed by the association of the variable portion of a light chain with the variable portion of the heavy chain. So assembly of antibodies from different combinations of light and heavy chains allows a person to produce a tremendous variety of antibodies from a modest number of different polypeptides. So when we take a closer look at the polypeptides of immunoglobulin reveals that variable portions of both heavy and light chains contain subregions that are especially variable or hypervariable from one antibody to another. Label HV. So light and heavy chains both contain three hypervariable structures that are clustered at the ends of each arm on the antibody molecule. As might be expected, the hypervariable regions play a prominent role in performing the structure of the antigen combining site, which can range from a deep cleft to a narrow groove or a relatively flat pocket. Deep cleft or a flat pocket. So, variations in the amino acid sequence of hypervariable regions account for the great diversity of antibody specificity, allowing these molecules to bind to antigens of every conceivable shape. The combining site of an antibody has a complementary stereochemical structure to a particular portion of the antigen, which is called an epitope. A combining site of an antibody has a complementary stereochemical structure to a particular portion of antigen, which is called epitope. That is an antigenic determinant. Because of their close fit, antibodies and antigens form stable complexes, even though they are joined only by non-covalent forces that individually are quite weak. The precise interaction between a particular antigen and antibody, as determined by X-ray crystallography, is shown. The two hinge regions within the molecule provide the flexibility necessary for the antibody to bind to two separate antigen molecules or to single molecule with two identical epitopes. Whereas the hypervariable portions of light and heavy chain determine an antibody's combining site specificity, the remaining portions of the variable domains provide a scaffold that maintains the overall structure of the combining site. The constant portion of antibody molecules are also important. Different classes of antibodies IgA, IgD, IgE, IgG and IgM have different heavy chains whose constant regions differ considerably in length and sequence. These differences enable antibodies of various classes to carry out different biological effector functions. So, for example, heavy chains of an IgM molecule bind and activate one of the proteins of the complement system, which leads to the lysis of bacterial cells to which the IgM molecules are bound. So, heavy chains of IgE molecules play an important role in allergic reactions by binding to specific receptors on the surface of mast cells, triggering in the release of histamine. So, in contrast, heavy chains of an IgG molecule bind specifically to the surface receptors of macrophages and neutrophils, inducing these phagocytic cells to ingest the particles to which the antibodies are bound. The heavy chains of IgG molecule are also important in allowing this class of antibodies to pass from the blood vessels of a mother to those of a fetus during pregnancy. While this provides the fetus and newborn with passive immunity to infectious organisms, it may cause a life-threatening condition called erythroblastosis fetalis. For this condition to occur, an Rh negative mother must have given birth to a child with an Rh positive phenotype, Rh positive Rh negative genotype during a previous pregnancy. The mother is usually exposed to the Rh positive fetal antigen during delivery of the first child who is not affected. If, however, the mother should have a second Rh positive pregnancy, antibodies present in her bloodstream can enter the fetal circulation and destroy the red blood cells of the fetus. Babies with this condition are given a blood transfusion either before, that means intrauterine, or after birth, which cleans the blood of maternal antibodies. Then some other aspects related to DNA rearrangements that produce genes encoding B and T cell antigen receptors. As discussed above, each IgG molecule is composed of two light L chains and two heavy chains. Both types of polypeptides consist of two recognizable parts, a variable portion whose amino acid sequence arises from one antibody species to another, and a constant portion whose amino acid sequence is identical among all H or L chains of the same class. What is the genetic basis for the synthesis of polypeptides having a combination of shared and unique amino acid sequences? In 1965, William Drainer of California Institute of Technology and J. Claude Bennett of the University of Alabama put forward the 2-gene 1 polypeptide hypothesis to account for antibody structure. In essence, Dreyer and Bennett proposed that each antibody chain is encoded by two separate genes, a C gene and a V gene, that somehow combine to form one continuous gene that codes for a single light or heavy chain. In 1976, Tonegua, I'm not sure about the pronunciation, working at a research institute in Basel, Switzerland, provided clear evidence in favor of the DNA rearrangement hypothesis. The basic outline of the experiment is very important. In this experiment, Tonegua and his colleagues compared the length of DNA between the nucleotide sequence encoding the C and V portions of a specific antibody chain in two different types of mouse cells, early embryonic cells and malignant antibody producing myeloma cells. The DNA segments encoding the C and V portions of the antibody were widely separated in embryonic DNA but very very close to each other in DNA obtained from antibody producing myeloma cells. These findings strongly suggested that DNA segments encoding the parts of antibody molecules become rearranged during the formation of antibody producing cells. So subsequent research revealed the precise arrangement of the DNA sequence that give rise to antibody genes. To simplify the discussion, we will consider only those DNA sequences involved in the formation of human kappa light chain, which are located on chromosome 2. The organization of sequences in germline DNA, the DNA of sperm or egg, that are involved in the formation of human kappa light chain is shown. In this case, a variety of different VK cells are located in a linear array and separated from a single CK gene by some distance. So nucleotide sequence analysis of these V genes indicated that they are shorter than required to encode the V region of the kappa light chain. The reason became clear when other segments in the region were sequenced. The stretch of nucleotides that encode the 13 amino acid at the carboxyl end of a V region is located at some distance from the remainder of the VK cell gene sequence. So this small portion that encode the carboxyl end of the V region is termed the J segment. There are 5 distinct JK segments of related nucleotide sequence arranged in tandem. The cluster of JK segments is separated from the CK gene by an additional stretch of over 200 nucleotides. A complete kappa V gene is formed, and as a specific VK gene is joined to one of the JK segments with the intervening DNA excised. This process is catalyzed by a protein complex called VDJ recombinase. So the VK gene sequence generated by this DNA rearrangement is still separated from the CK gene by more than 2000 nucleotides. No further DNA rearrangements occur in kappa gene assembly prior to transcription. The entire genetic region is transcribed into a large primary transcript from which introns are excised by RNA splicing. So rearrangement begins as a double-stranded cuts are made in the DNA between a V gene and a J gene. The cuts are catalyzed by a pair of proteins called RAG1 and RAG2 that are the working pairs of the VDJ recombinase. The 4 free ends that are generated are then joined in such a way that the V and J coding segments are linked to form an exon that encode the variable region of the polypeptide chain, while the 2 ends of the intervening DNA are linked to form a small circular piece of DNA that is displaced from the chromosome. So joining of the broken DNA ends is accomplished by the same basic process used to repair DNA strand breaks that are depicted. Then the rearrangement of IgDNA sequence has important consequences for the lymphocyte. Once a specific VK sequence is joined to a JK sequence, no other species of kappa chain can be synthesized by that cell. It is estimated that the DNA of human germ cells contains approximately 40 functional VK genes. Thus, if we assume that any V sequence may join to any J sequence, we expect that a person can synthesize approximately 200 different kappa chains. But this is not the only source of diversity among these polypeptides. The sites at which the J sequence is joined to a V sequence can vary somewhat from one rearrangement to another, so that the same VK and JK genes can be joined in 2 different cells to produce kappa light chains having different amino acid sequence. So additional variability is achieved by the enzyme deoxynucleotidal transferase, which inserts nucleotides at the site of strand breakage. These sources of additional variability increase the diversity of kappa chains an additional tenfold, bringing the number to at least 2000 species. The site at which V and J sequence are joined is part of one of the hypervariable region of each antibody polypeptide. Thus, slight differences at a joining site can have important effects on antigen-antibody interaction. We have restricted our discussion to kappa light chains for the sake of simplicity, but if you need to learn some other aspects, I encourage you to read that. So, similar type of DNA rearrangements occur during the commitment of a cell to the synthesis of a particular lambda light chain and to a particular heavy chain. Whereas the variable regions of light chains are formed from two distinct segments, V and J segments, the variable regions of heavy chains are formed from three distinct segments, V, D, and J segments, by similar types of rearrangement. DNA rearrangement of heavy chain locus precedes DNA rearrangements of the light chain locus. The human genome contains 51 functional VH segments, 25 DH segments, and 6 JH segments. Given the additional diversity stemming from the variability in VH, DH, and DH, JH joining, a person is able to synthesize at least 1 lakh different heavy chains. The antigen receptors of T cells, TCRs, also consist of a type of heavy and light chain whose variable regions are formed by a similar process of DNA rearrangement. The formation of antibody genes by DNA rearrangement illustrates the potential of the genome to engage in dynamic activities. Because of this rearrangement mechanism, a handful of DNA sequences that are present in the germ line can give rise to a remarkable diversity of gene products. So, as discussed above, persons synthesize roughly 200 different species of kappa light chains and 1 lakh different species of heavy chains. If any kappa light chain can combine with any heavy chains, a person can theoretically produce more than 200 million different antibody species from a few hundred species genetic elements present in the germline. So we have see how antibody diversity arises from the presence of multiple V exons, J exons, and D exons in the DNA of a germline, variability in VJ and VDJ joining, and the enzymatic insertion of nucleotides. An additional mechanism for generating antibody diversity, referred to as somatic hypermutation, occurs long after DNA rearrangement is complete. When a specific antigen is reintroduced into an animal following a period of time, antibodies produced during the secondary response have a much greater affinity for the antigen than those produced during the primary response. So, increased affinity is due to small changes in amino acid sequence of variable regions of the heavy and light antibody chains. These sequence changes result from mutations in the genes that encode these polypeptides. It is estimated that rearranged DNA elements encoding antibody heavy regions have a mutation rate 10 raised to 5 times greater than that of other genetic loci in the same cell. So, included in the mechanism responsible for this increased level of V region mutation is an enzyme known as activation induced cytosine deaminase AID that converts cytosine residues in DNA into uracil residues, leading to UGE mismatches and subsequent mutations during DNA repair and one or more translation DNA polymerase that tend to make errors when DNA containing uracil is copied or repaired. So, persons who carry mutations in AID and unable to generate somatic hypermutation are plagued by infections and often die at an early age. So, somatic hypermutation generates random changes in the V regions of Ig genes. Those B cells whose genes produce Ig molecules with greater antigen affinity are preferentially selected following antigen re-exposure. So, selected cells proliferate to form clones that undergo additional rounds of somatic mutation and selection, whereas non-selected cells that express low affinity Igs undergo apoptosis. In this way, the antibody response to recruit or chronic infections improves markedly over time. Once a B cell is committed to form a specific antibody, it can switch the class of Ig it produces from IgM to IgG by changing the heavy chain produced in the cell. This process, known as class switching, we will be learning later. Class switching is an important term. Occurs without changing the combining site of antibody synthesized. Recall that there are 5 different types of heavy chains distinguished by their constant regions. The genes that encode the constant regions of heavy chain are clustered together in a complex. So class switching is accomplished by moving a different CH gene next to the VDJ gene that was formed previously by DNA rearrangement. Class switching is under the direction of cytokines secreted by helper T cells during their interaction with the B cell product producing the antibody molecule. For example, the helper T cell that secretes IFN gamma induces a switch in the adjacent B cell from its initial synthesis of IgM to synthesis of one of the IgT classes. Class switching allows the lineage of B cells to continue to produce antibodies having the same simplicity but different effector functions.